This is Lecture 12, Emotional and Motivated Behavior, and it corresponds to Chapter 12 of your book. Few reminders and announcements. Homework 13 can be turned in until 12:15, so this is a bit of an extension. Exam 3 will post this weekend, and it'll cover chapters 10 through 14. Technically, it's due on 12:15, which is the end of finals week. However, I don't need to turn in grades until 12:19, and I can extend it to 12:17 if anyone needs extra time. So please contact me at least a few days beforehand if you would like a few extra days to complete the final.
Reading recommendations, these are not required. This is sort of related to the language section. There's this poem called The Lion-Eating Poet in the Stone Den that is in Mandarin Chinese, and I don't speak Mandarin, but my understanding it's different versions because Mandarin is a tonal language. It's different version of the shi syllable, and it's all different versions that actually tell an entire story using this one syllable. And as well, there was a recent article on dreaming in Nature Neuroscience that some people might find interesting. Please do keep in mind that news articles, even in really good newspapers, almost always overstate the significance of new studies. So I can't tell you how many times I've read an article that said it was revolutionizing memory or sleep research or schizophrenia or depression research. Just always take that information with a grain of salt. And then, this is also not required, but some people were interested in that fatal familial insomnia that was mentioned in the previous chapter, so here's a bit more information on it if you like. I'll review it really quickly. So fatal familial insomnia is autosomal, which means it's not sex-linked. It's on one of the non-sex chromosomes, and it's a dominant genetic disorder. It's caused by the specific gene, the PRNP gene, which codes for prion proteins, and this is a similar, prion proteins are also implicated in mad cow disease, if anyone remembers that outbreak a number of years ago. The gene is involved in the sleep-wake cycle, memory, and motor function. The disorder actually usually shows up around middle age, and I couldn't find information why exactly that is. It's very, very rare. I think there's only like maybe 40 families that have this gene that are on record. And there are 4 stages. So there's first, insomnia becomes worse, and this is accompanied by daytime issues such as anxiety, panic attack, and paranoia that lasts about 4 months. Then there's an increase in panic attack and onset of some hallucinations. This lasts about 5 months. In the third stage, sleep is completely impossible, so the subject completely stops sleeping, and this lasts about 3 months. And then finally, for about 6 months, symptoms of dementia set in. Patient really doesn't have any cognitive or motor functioning. They're not often not very conscious at all, and this results in death. Towards the end of the disease, the patient actually can be trapped in hypnagogia, which is a state right before falling asleep. So we talked about hypnagogic hallucinations earlier, which is maybe when you're in that stage right between sleep and awake, where you might hear somebody calling your name or feel like you had the sensation that you fell down, something like that. This state between wake and sleep is hypnagogia. And at the end of fatal familial insomnia, people can get stuck in that state until they eventually pass on. There's no effective treatment for this illness yet. Sleeping pills can actually make the symptoms worse. But I found one recent clinical trial with doxycycline, which is an antibiotic and actually an anti-malarial. So if you go to a place that has malaria, such as certain regions of Africa, they will prescribe you doxycycline actually as a prophylactic against malaria. Then the same medication, this antibiotic, shows some promise in clinical trials of at least slowing down the symptoms. This whole syndrome kind of brings up an interesting point about treatment research, which is something that's present also in other, especially like dominant or sort of single gene diseases, doing research with people who have these fatal genetic disorders. You will typically have like a family, as many people as you can from a family participate, and some people in the family don't want to know if they have the fatal genotype, and the scientists, of course, for the sake of research, have to know whether this individual has the fatal genotype. And so there's kind of a range of interesting ethical issues that come up when scientists have this information about an individual's likely future, and in this case, whether or not they're likely to have a fatal disease in middle age. Okay, so moving on to chapter 12, this is section 1, identifying the causes of behavior. This the section is about emotion and motivation, and emotion and motivation have a lot in common, and therefore we're kind of covering both of these in the same chapter. Emotion is a complex pattern of changes, including physiological arousal, feelings, cognitive processes, as well as behavioral reactions that are made in response to a situation perceived to be personally significant. This is the APA definition of emotion. And then motivation is the process of starting, directing, and maintaining physical and psychological activities, including mechanisms involved in preferences for one activity or another, as well as the vigor and persistence of responses. So you can see by these definitions that both of these are kind of broad constructs, broad topics that cover a lot of different processes. There are many different causes of behavior, of course. Our brain is specifically designed for certain behaviors to be naturally rewarding, and these are typically things that help us to survive. So food, sex helps us procreate, exercise keeps us healthy and strong, and our brain is naturally designed for that to be rewarding. The brain as well craves stimulation, so we are naturally curious, especially in our younger stages, like kids, babies are naturally curious. Sensory deprivation, having no stimulation, is very aversive to us. One of the worst punishments you can give an individual is long-term solitary confinement. The absence of stimuli actually often makes our brain create its own stimulation. So people in solitary confinement, especially if it's dark in there, in the least humane contexts of solitary confinement, people have hallucinations. The brain starts making up stimuli, as well as having heightened senses, so maybe having a really strong sense of smell in the absence of visual stimulation. Reward circuits in the brain are activated by both endogenous or inside stimuli that comes from inside your own brain, such as hormones and neurotransmitters, as well as thoughts and feelings. So thinking about something can be rewarding. Thinking about someone that you're in love with can be a rewarding activity in itself. Reward circuits can also be activated by exogenous or outside stimuli, such as sights and sounds and smells from the environment. Moving on to section 2, the chemical senses. The chemical senses include olfaction, which is the sense of smell, and gustation, the sense of taste. They are called chemical senses because the environmental stimuli interact directly with the receptors in these two senses. They appear to be less complex in general than other sensory systems, so we are going to cover it in a little less detail. But there's also a lot we don't know, especially about olfaction. And so there's certainly the paucity of information that we have may not actually represent the full complexity of the system. Olfaction in general is more primitive, and it's highly connected with emotion and memory and motivation as well. So that's why we're covering the chemical senses in the motivation and emotion chapter, is because these senses are intimately involved with emotion and motivation. So olfactory receptor neurons interact directly with substances from the environment, with the chemicals that come in through the air. Olfactory epithelium, which is this layer of tissue, has receptors and support cells embedded within it. The receptor cells have 10 to 20 cilia, which protrude from the olfactory mucosa. This is kind of like mucus in the olfactory system. Scent chemicals dissolve in this olfactory mucosa which then connect with the olfactory sensors or receptors that are specialized to respond to a different range of scents. So just like how different rods and cones in the retina respond to different light stimuli or different aspects of light stimuli, olfactory receptors specialize in different scents, different types of chemicals. Odors activate a pattern of specialized receptors, and then the summed activation of these codes for a given scent in the brain. Olfactory pathways in the brain. So this goes from olfactory receptor cells to the olfactory bulb, which is the main olfactory area in the brain. Olfactory receptor cells create balls of dendrites, and these synapse with something called mitral cells. Mitral cells send their axons to forebrain areas, in particular the amygdala. So the olfactory system is very highly connected with the amygdala. Amygdala we'll talk about a bit more later, and we've talked about in the past, but amygdala is responsible for a number of things, including emotional memory and the valence of emotional response, fear response, etc. The piriform cortex, which is also called the primary olfactory cortex, also gets projections from the mitral cells. This is, interestingly, a trigger zone for some people with epileptic seizures. So some people who have epileptic seizures have what's called an olfactory aura before the seizure, which means that they smell something that actually doesn't exist. And they smell typically the same thing every time before the seizure starts. So there are examples of people always smelling oranges before they have a seizure, and this is called an aura, olfactory aura. Mitral cells also send axons to the thalamus, which, if you remember, is kind of the switchboard in the brain. Pretty much all of the sensory systems and most of the sensory pathways go through the thalamus before that information is sent to other areas of the brain. So in the case of the olfactory system, information goes from the thalamus to the orbitofrontal cortex, which is responsible for sensory integration, emotion, and reward in decision making. And we're going to talk more about the orbitofrontal cortex a little bit later in this section. Pheromones are biochemicals that are released by animals and act as a signal. So people typically know about pheromones because they are involved in a lot of animal sexual behavior, but pheromones are also involved in bonding between the mother and the child, as well as feeding between the mother and child specifically, so breastfeeding. The mother gives off certain pheromones that allows the baby to smell the mother and locate, and also encourages breastfeeding. There's information in your book about the female menstrual synchrony and how the chemical senses may be involved in this. Be really wary of research on female menstrual synchrony. The existing research is troubled with methodological issues. One, it does happen that women become synchronized, but there isn't strong indication that it's at above chance. So if women are menstruating for 5 days out of the month, there's at least some percentage of chance that those menstrual cycles are going to overlap. So in my opinion, I think female menstrual synchrony sounds like a bit of a myth. And there's indication that it's not caused by pheromones necessarily, but more likely that women living together in the same environments are engaging in a lot of the same environmental habits. So similar sleep cycles, similar eating, and other environmental influences. That if we do perceive, or if menstrual cycles do synchronize, it may be more due to these environmental factors than pheromones. Also interesting in terms of the olfactory system, people can identify their own odor. So if they smell their own odor outside of themselves, they know that that's their own odor, even though most of the time we're not aware of our own odor. They can also identify family members and friends, the odor of family members and friends above chance. Women prefer the scent of genetically different men, which we presume to be adaptive in an evolutionary sense. It's better for people who are genetically different to have offspring together. It kind of increases the immune fitness of their offspring. And finally, just an interesting bit of another interesting study where men smelling women's tears had reduced sexual arousal and reduced testosterone levels, the two of which are correlated, and did not appear to have increased empathy in smelling these tears. So the scent of women's tears actually had an effect on men, and I don't know about the effect of men's tears on women, but kind of speaks to the power of the olfactory system. Moving on to gustation. Different species have different taste preferences, typically based on what types of foods they need for survival. Humans prefer sweetness. This may be because we were hunter-gatherers previously and ate a lot of fruit. Within humans, or among humans, there's a lot of variation in taste, so different people prefer different tastes and also are better at discerning different tastes. So some people, sweet tastes a lot sweeter, for example, to some people compared to others. There are 5 basic tastes: bitterness, sweet, salty, sour, and umami, which is like MSG. Other features that the tongue can sense that are not typically considered taste are spiciness, which is actually a form of pain, temperature, mouthfeel or fattiness, and metallicness. These are not detected by taste buds, but they're detected by other sensory receptors in the mouth and on the tongue. So taste receptors are located on taste buds, and they're on the tongue, under the tongue, on the soft palate or the roof of the mouth, the back of the mouth, and then even in the nasal cavity located above the mouth. Taste buds themselves are located on these little bumps called papillae. They last for an average of 10 days. Each taste bud lasts for an average of 10 days, so a really high turnover rate for sensory receptors. There are individual receptors for each of the 5 tastes. The tongue taste map is a myth. It's not true. So there's this myth that I think is pretty pervasive that, for example, sweetness is tasted on the tip of the tongue, where bitterness is tasted exclusively in the back of the tongue. Sour and salt are on the sides. It is true that there are certain parts of the tongue that are slightly more sensitive to certain tastes, but taste can be detected, all the tastes can be detected on all parts of the tongue. Chemicals from food dissolve in saliva and interact with microvilli, which opens up ion channels, and this is how taste is transferred into neural signals. There are 2 different gustatory pathways in the brain. So both of them start with the taste bud, which synapses with afferent cranial nerves, 7, 9, and 10, and this forms the main gustatory tract into the brain. It's an afferent tract. If you remember that sensory information is afferent, meaning it's heading toward the brain. When it enters the brainstem and then it splits, one track goes through the thalamus, this switchboard for sensory information, and then that splits and goes to the primary somatosensory cortex and then the insula, which is the primary gustatory cortex. The insula tract then sends projections to the orbitofrontal cortex, which is where the perception of flavor is. Orbitofrontal cortex is part of the prefrontal cortex. Another tract after the brainstem split goes to the amygdala, which is again responsible for valence of emotion, emotional memory, and the hypothalamus. Hypothalamus controls basic feeding, sleep, emotional functions. We'll talk more about the hypothalamus in some sections that are coming up. Taste is highly affected by expectation. So if people expect to taste something, they're more likely to taste it. Wine taste is affected by the price. People think that higher priced wine tastes better, even though in a meta-analysis of blind wine tastings, non-experts, so not sommeliers, but just kind of lay people, preferred less expensive wine, actually slightly more than the expensive wine. So there's a lot of indication that price differences in wine may not actually reflect people's baseline taste preferences. But a lot of things, other things in wine, like color of wine, makes us more likely to taste one way or the other. They've done studies where they have wines that are like a red wine and tell people it's white but don't show them the color, and people are more likely to believe that it's white wine. The ambiance of the room, the descriptions. So if you've done a wine tasting, they might say you'll find, you know, nutty notes or hints of lemon or something like that. And you're more likely to taste these if somebody tells you that that's what you're looking for. There are people called super tasters, and these are people who have a higher perception of taste than others. Very, very good sense of taste. It's relatively common, actually. It may be one reason that some people are picky eaters. It's slightly more likely in women, also slightly more likely in people from Asia, South America, and Africa. Super tasters actually have more taste buds, so they actually have more sensory receptors. There are also people called non-tasters who have fewer taste buds and do not have a very good sense of taste at all. Bitterness, it specifically has very strong genetic influences, and a version of bitterness may be adaptive. Actually, there are many toxic substances that are bitter. For example, young leaves are less bitter than old leaves, and old leaves contain more toxic substances. For one example of this, little kids tend not to like bitter foods as much, and little kids are also more susceptible to toxic substances. Other genetic influences on taste include coriander, or cilantro. Cilantro and coriander are the same plant. Some people really, really hate coriander and cilantro, and believe that it smells and tastes like soap. And there's actually a genetic variant that is linked to this perception. Some people feel really, really strongly about their hatred of cilantro. Apparently there's this website where people write haikus to share with other members of this cilantro hating club. Okay, so moving on to section 3, evolution, environment, and behavior. Evolutionary psychology is a branch of psychology that applies principles of natural selection to human behavior. So we've kind of done a lot of that throughout this course, trying to look back and see why the brain might have evolved the way it did, why behavior might have evolved the way it did in humans. The caveat to evolutionary psychology is that it's difficult, maybe even impossible, to test the hypotheses of evolutionary psychology experimentally. We can never really know why we evolved the way we did. We can't go back in time and test these things. So we're kind of piecing things together from a lot of clues and making a lot of inferences or best guesses. One aspect of the evolution, or one thing that we think we've been able to discern from evolutionary psychology, is that there is this mechanisms that we call innate releasing mechanisms in the brain, and these exist in the human brain and the animal brain, and they're old mechanisms that are triggered by specific stimuli that trigger more specific responses. They're innate, they're not learned by experience, and they're present at birth. And so these are kind of, you might call them instinctual behaviors. For example, in cats, if they're feeling defensive, if they see maybe a dog, they'll arch their back and bare their teeth in a very predictable way, and even kittens will do this. Innate releasing mechanisms in humans include emotional facial expressions. So across all different cultures and all different types of people, the facial expressions for a lot of the basic emotions, so happiness, sadness, anger, surprise, are all the same. And we see them in babies who haven't had time to really learn imitation yet, who are not at the level of imitating yet. Congenitally blind babies also show the same expressions, which I think is very convincing. So babies who have never seen facial expressions before still show this facial expression of surprise when they're surprised by something, happiness, etc. An example of this is babies trying lemons. This is actually just really cute. So babies who try lemons all seem to make the same face in response to very sour things. So you can follow that YouTube link for a compilation of different babies showing a similar reaction to lemons. Mate preferences also appear to be evolutionarily guided. Women tend to prefer earning potential power and education more than men prefer that, which people have suggested may be due to gender roles that that existed kind of throughout the evolution of humans, where women wanted a man who is likely to be able to support offspring. They also tend to prefer higher waist to chest ratios, which is also associated with testosterone and possibly higher fertility. Men tend to prefer good looks in women, health, and tend to prefer women who desire family. They also tend to physically prefer a higher waist to hip ratio in women, which is also an indication of a biological indication of fertility. So there tend to be characteristics of mates that males and females prefer that may be based on evolutionary advantages in relative mates, in relative sexes. Moving on to section 4, the neuroanatomy of motivated and emotional behavior. The most important structures in the brain for motivated and emotional behavior are the hypothalamus and the related pituitary gland, the limbic system, and the frontal lobes. And this includes the reward pathways within these systems. The hypothalamus controls many systems for basic and automatic behaviors, and the frontal and limbic systems project into the hypothalamus. They kind of send information to the hypothalamus to create motivated behaviors. There are two main types of motivated behaviors: regulatory and non-regulatory behavior. Regulatory behavior is motivated by individual survival needs, so maintaining body temperature, eating, drinking, waste elimination, things we need to do day-to-day to survive. And these regulatory behaviors are maintained by homeostatic mechanisms, which are processes that keep certain body functions in a critical narrow range of function. And these homeostatic functions are regulated by the hypothalamus. Hypothalamus is a pretty small brain structure that does quite a lot of different functions, or at least regulates a lot of different functions. So regulatory behaviors work to maintain a homeostatic set point, this ideal range of functioning. An example would be 37 degrees Celsius body temperature that our body tries to maintain within a pretty limited degree of functioning. If it gets too high, we have a fever and our body starts to break down. So deviations from the set point typically cause behavior to fix the deviation. So for example, if we have a fever, we start sweating so that to cool off our skin. If our body temperature is too low, we start shivering to bring up the body temperature. Non-regulatory behaviors are not necessarily motivated by basic survival needs. So this includes sex, parenting, curiosity, so exploring the environment around us, aggression, and food preferences. Some non-regulatory behaviors are mediated by the hypothalamus, such as sex, and others are not. So curiosity is not driven by the hypothalamus. More frontal lobe features are frontal lobe, which is always involved in higher order cognition, or at least the prefrontal lobe is involved in higher order cognition. So the frontal lobe is more involved in non-regulatory behaviors as compared to regulatory behaviors. The hallmark of regulatory behaviors is that they are mediated strongly by the hypothalamus. The hypothalamic circuit is responsible for regulatory functioning. This influences the endocrine system, which is the hormone system, and the autonomic nervous system, as well as limbic system functioning. The hypothalamic influences on the hormones are mediated by three main regions of the hypothalamus: the lateral region, medial, and paraventricular. However, we often talk about hypothalamus as in terms of the nuclei, or the really small specific parts of the hypothalamus that's responsible for a given function. In this lecture, I'm probably not going to talk too much about specific areas of the hypothalamus because we can get really detailed. There are some highly specialized functions in different areas, so I'm going to try to keep it pretty broad in terms of the subdivisions of the hypothalamus. The medial forebrain bundle is one of the most well-known hypothalamus tracts. It's actually a tract that runs through a number of structures. It's the main white matter track that runs through this ventral section of the brain. Axons in the medial forebrain bundle project to and from the hypothalamus, so the projections go in both directions. It connects brainstem structures to the limbic system, also connects the brainstem to basal ganglia and frontal cortex, and a lot of these, the track is predominantly dopaminergic, so dopamine neurons are making these connections. The medial forebrain bundle is involved in eating, sexual behavior, addiction, impulsivity, a lot of the things that are mediated by the hypothalamus. So, as I mentioned before, different regions or nuclei, groups of neurons in the hypothalamus control different behaviors. These different regions also use different neurotransmitters. The hypothalamus is intimately connected with the pituitary gland. It is kind of the master of the pituitary gland. It sends directions to the pituitary. For example, the paraventricular hypothalamus produces vasopressin and oxytocin, which we talked about briefly before in the hormone section. These are released by the posterior pituitary gland, which is considered by some to be like an extension of the hypothalamus. It shares a lot of the same tracks. Oxytocin is responsible for child-parent bonding behaviors, breastfeeding, prosocial behavior, sexual motivation, pair bonding. Vasopressin is also implicated in some of these functions, as well as water retention in the kidneys and blood pressure. The pituitary gland is, as I mentioned before, this endocrine gland, this part of the hormone system attached right below the hypothalamus. And you can see that a lot of its projections are really straight to the hypothalamus. So these two structures are very intimately connected. The hypothalamus sort of tells the pituitary gland what to do. It controls the release of hormones from the pituitary. There are two main regions of the pituitary, the front and the back, which is known as the anterior pituitary, the posterior pituitary. You can see in this chart below some of the main hormones that are produced by the anterior pituitary. The control of hormones that is released by the hypothalamus, or the control of hormones through the pituitary by the hypothalamus, is mediated by three main factors. The first one is feedback loops. So these are receptors in the hypothalamus detecting levels of hormones and then releasing or inhibiting the release of hormones. So if there's too much of a hormone or too little of a hormone, this information is coming back to the hypothalamus from the body, telling it, give us more or stop releasing hormones. There's neural control, so things that are happening in higher level brain areas, such as anxiety, visual stimuli, these can influence the release or inhibition of hormones as well. And then there's experiential responses, and this is the fact that hormone activities can structurally change the hypothalamus over time, creating systematic increases or decreases in hormone release. So somebody can be more likely to release certain hormones based on life experiences, and how their hypothalamus has been formed throughout their life. The limbic system involves many areas of the brain, and it's in this medial subcortical section of the brain mostly. Different people define the limbic system slightly differently. So, for example, your book includes the sensory association cortex within the limbic system. Some people don't. But the main actors are the thalamus, the hypothalamus, the amygdala, the hippocampal formation or the hippocampus, mammillary nucleus, and the prefrontal cortex, and the cingulate cortex in the medial section of the brain as well. Limbic system has a lot of functions, including emotion, motivation, memory, olfaction, so a lot of really important functions. Amygdala is one of the main actors within the limbic system. This is plays an important role in emotion and species-typical behaviors. Specifically, the amygdala is associated with valence of emotion, or the strength with which somebody feels an emotional experience. Highly associated with fear and fear conditioning, anxiety, as well as eating and sexual behavior. Stimulation of the amygdala in both humans and animals creates fear behavior in animals and subjective experience of fear in humans. It also increases the blood pressure, so it communicates with the hypothalamus to increase the blood pressure. Removal of the amygdala creates the loss of fear, increased and indiscriminate sexual behaviors, overreaction to visual stimuli, so kind of just overreacting emotionally to things in the environment, as well as, interestingly, visual agnosia, which, if you remember, is an inability to recognize objects, visual objects. So the amygdala obviously plays a lot of really important functions. It plays a lot of important roles within the human system. Olfactory system connects directly to the amygdala in humans and in other animals, so sense of smell and emotion are intimately connected, as well as sense of smell and fear are intimately connected. The HPA axis is a system in the brain and body that actually wasn't mentioned in your book, but I think it's important to know about, especially in motivated and emotional behavior. It stands for the hypothalamic pituitary adrenal axis, and it regulates stress reaction in humans, the basic stress reaction. So it starts at the paraventricular nucleus of the hypothalamus, which creates and secretes corticotropin-releasing hormone, or CRH, as well as vasopressin, which, if you remember, vasopressin can increase blood pressure. That sends the CRH to the anterior lobe of the pituitary gland, and these hormones stimulate secretion of adrenocorticotropic hormone, ACTH. The adrenal gland, specifically the adrenal cortex, produces cortisol and other glucocorticoids, as well as epinephrine, norepinephrine, in response to the hormones that have come through this system. And that has an effect on a lot of different parts of the body, but it's essentially a reaction to stressful things in the environment, a way to produce cortisol. Cortisol is like a stress hormone, so it's going to increase blood pressure, increase heart rate. It stops other functions in the body, so it stops digestion, and it increases the subjective feeling of anxiety and stress. And there's a negative feedback cycle in this as well, where if there's a lot of cortisol in the body, the hypothalamus will receive feedback about this and inhibit activity of the HPA axis. The HPA axis has been implicated in a lot of different disorders, like anxiety disorders, phobias, PTSD, even depression. So I think it's really important if you're interested in clinical applications of this information. And if you like, there's a pretty good video that does like a 2-minute explanation of the HPA axis that has animations. The prefrontal lobe has a number of functional subdivisions. The orbitofrontal cortex is one of the main divisions. It has a lot of connections to the amygdala and the hypothalamus. It's responsible for a number of functions, including delayed gratification, planning the appropriate social responses, as well as suppressing inappropriate responses within the social and emotional realms. People with lesions to the orbitofrontal cortex have emotional dulling, lack of motivation. They'll act inappropriately, especially sexually or socially, and have difficulty planning their day, getting out the house because it's hard to do everything, hard to plan everything, to even like go out to the grocery store, for example. In the 50s, there was an unfortunate period of time during which transorbital lobotomies, or ice pick lobotomies, were popular for people who had behavioral problems or severe psychiatric illnesses. So scientists had noticed that the frontal lobe was associated with regulation of behavior, and they thought by lesioning or lobotomizing that part of the brain, they could fix some of this behavior. But they were rather cavalier about it. and it was a pretty brutal procedure. Literally, a tool that looks like an ice pick, it's a medical tool, was inserted through the eye socket. Sometimes the patient was awake, and literally was just wiggled around to lesion the orbitofrontal cortex, and resulted in a lot of severely disabled people, essentially just vegetables in some cases, or people who had even less behavioral control after this. It was particularly unfortunate for people with schizophrenia and other severe psychiatric illnesses. A lot of people ended up with even more severe disabilities due to these lobotomies. There's also the ventromedial prefrontal cortex, which is functionally pretty similar to the OFC, and it's also right next to the OFC, just more medial. It's responsible for inhibition of emotional responses, decision making, and possibly empathy and sympathy. Damage to this area of the brain during childhood can result in antisocial behaviors and poor moral judgment, leading some people to say that this is kind of the moral center of the brain. And then there's the dorsolateral prefrontal cortex. It's one of the most recently evolved areas of the brain, and it's associated with the highest level of cognitive functioning. So this is like the CEO of the CEOs. It's responsible for abstract reasoning, inhibition again, planning, decision making, and working memory. It's activated during complex social interactions, including theory of mind, which is understanding what another person might be thinking, as well as the suppression of selfishness. And the sense of justice that humans typically feel may originate in the dorsolateral prefrontal cortex. There's a researcher, he's a primatologist at Vanderbilt, who name's Frans de Waal, and he is interested in emotions and kind of what we think of as human emotions and human capacities, and whether or not these are seen in primates as well. And he has this really great video showing an experiment in which capuchin monkeys appear to have a sense of injustice just like humans, and I highly recommend you watch this video. So moving on to emotions, stimulating and expressing emotions, there are a lot of people have argued that there are 6 basic emotions. This originated with this guy, Paul Ekman. Other people say there are 4 or up to 10. There's also dimensional versions of emotions where the basic emotions can kind of be experienced in a range of degrees of that emotion. So the 6 basic emotions that Ekman proposed, and most other theories at least include most of these emotions are anger, disgust, fear, happiness, sadness, and surprise. Emotions are typically experienced on 3 different levels. A physical level, so emotions are typically heart rate might change, blood pressure might change, body temperature might change. They can be experienced through subjective experience, just the feeling of being happy or angry. And then the cognitive appraisal of the emotions. So when we recognize what I'm feeling right now is happiness, I'm feeling happiness because this good thing happened to me. There are a number of theories about the experience of emotion and the relationship between these 3 levels of the experience of emotion. These were likely covered in your Psych 100 class. I'll go over them very briefly. The James Lange theory says that physical symptoms of emotions come first, and then cognitive appraisal of those symptoms, and this results in a subjective experience of those emotions. So the brain creates an emotional explanation for physiological conditions. An example of this would be seeing a bear triggers your heart rate to increase, and then we interpret this experience as fear. We know we're supposed to be afraid and we see a bear, so we're saying I'm feeling fear right now. The Cannon-Bard theory, it's not in opposition to the James Lange theory, it kind of adds on to it. Cannon and Bard noted that the subjective experience of emotion happens quickly. It happens too quickly to actually be the result of cognitive appraisal. We just don't have time to go through that thinking process before we feel that emotion. So Cannon and Bard felt that emotional and physical responses to the evoking stimulus, the bear, are separate and independent. As is the case in most somewhat opposing theories, although these aren't in direct opposition. The truth is probably somewhere in between a two-factor theory, like a combination of James Lang and Cannon-Bard, where to some extent physical emotions come first and then we interpret them, but there's also physical separate processes happening as well. Evidence for the physiological sensations influencing emotion. There's a lot of evidence for this. Notably, the same physiological conditions underlie a range of emotions. So an increased heart rate can indicate fear, anger, or excitement. But evidence that physiological sensations do influence emotion, it mainly comes from the incapacitation of the ability to react physically. So when you can't physically react in the way that's associated with emotion, you have a decreased experience of that emotion. For example, the degree of paralysis. So you can have paralysis that's only your legs, or all the way up to quadriplegic, which includes all four limbs. And the degree of paralysis is associated with loss of emotionality. So the more paralyzed somebody is, the less they're feeling emotions. As well, inhibition of facial movement associated with an emotional expression is associated with a less emotional experience, and inducing the facial expression without without the actual emotional stimulus is associated with a more positive mood. And these latter two are demonstrated on the right here. So in the first one, this black and white picture of the woman, she is holding a pen in her mouth in such a way that she cannot smile, actually. It's inhibiting smile muscles. And in a study, this was associated with a lower mood, but she was, as opposed to somebody who was not holding a pen in their mouth, who was holding something in their mouth in a different way that still allowed them to smile. In the second picture, there are studies indicating that somebody holding a chopstick or a pencil in their mouth in such a way that forces them to smile, such as these, what look like stewardesses are doing. This is associated with actually feeling better, feeling more happy. So moving on to control of regulatory behaviors, the control of eating. There are three main sources of the control of eating. This is the digestive system, which sends out signals, the hypothalamus, and cognitive factors as well. So the digestive system is, of course, controlled by the ENS, and it extracts lipids, amino acids, and glucose from food, glucose being the primary source of energy for the brain. Food interacts with receptors in the ENS to release peptide hormones, which in turn signal to the brain and other areas of the nervous system that somebody has had enough food, satiety. The hypothalamus is the main structure that's involved in feeding behavior in the brain. The stimulation of the lateral hypothalamus results in increased eating, whereas a lesion of it completely stops eating. Conversely, stimulation of the ventromedial hypothalamus decreases eating, whereas a lesion creates extreme overeating. So these two sections, these two nuclei within the hypothalamus, are actually highly specialized in their function in regards to eating, and these are results that have been replicated a number of times with rats, where you'll get an obese rat when you lesion this part of the brain that's associated with satiety, and what is essentially an anorexic rat, that when you lesion these other areas of the brain, the lateral hypothalamus. Notably, other regions of the hypothalamus are involved, but these are the main that have been studied and appear to be the main areas involved. And from human studies, we have found that there are abnormalities in the hypothalamus, both functionally and structurally, in disorders of dysregulated eating, which includes some types of obesity. Not all types of obesity are associated with people eating too much, at least not behaviorally. There can be a lot of factors that cause obesity, but in some individuals with some types of obesity, the hypothalamus is affected, as well in anorexia, this disorder where people don't eat enough food to the level that they're in physical danger, there have been consistent indications that the hypothalamus is affected. Cognitive control of eating. So eating in general brings people pleasure, even images, internal, so imagining a really nice juicy cheeseburger, or external, such as a picture of cookies, can trigger hunger. Learned associations make us more or less likely to enjoy a food. If your grandma made a certain type of food when you were a kid, you might have a learned association to make that more pleasurable to eat. On the other hand, food poisoning from a chicken sandwich, for example, might make you averse to chicken sandwiches. Damage to the amygdala creates an extinction of food aversions. So you essentially don't have like this fear or anxiety response to food that you might have been averse to for whatever reason previously. Damage to the orbitofrontal cortex can cause a decrease in eating, possibly due to if the orbitofrontal cortex is damaged, this might have lesioned the inputs from the olfactory bulb, and there might be a decrease in the association of reward associated with food and drink taste and smell. In terms of controlling drinking, there's two main types of thirst. Osmotic thirst, which is from high volumes of solutes in body fluids. So even if you have a lot of body fluids, if there's a lot of like salts or other solutes in those fluids, that's osmotic thirst. So the hypothalamus detects solute concentration and triggers thirst for water. The hypothalamus can say specifically you need water. In this case, it wouldn't say drink milk, for example. And then there's hypovolemic thirst, which is from loss of fluid volume in the body. And this actually is regulated by a different hypothalamic circuit, and it stimulates you to drink fluids with solutes. So you need electrolytes as well in this case. So it might stimulate you to drink like juice or milk or something, fluids that also have nutritive substances in them. In terms of controlling sexual behavior, this is highly controlled by sex hormones in the brain, and the sex hormones have two main ways of controlling the brain. It controls the developing brain and the adult brain in different ways. Effects on the developing brain are also called organizing effects because it sort of organizes how the brain, it organizes certain functions in the brain. Sexual dimorphism is physical differences between different sexes other than sex organs. There's sexual dimorphism in the brain and the nervous system. Women tend to have larger volumes in the limbic areas and frontal regions, whereas men tend to have larger volume in the hypothalamus and parietal regions. This is relative to brain size. Women do on average have a smaller brain size as well. The estrogen creates sexual differentiation in the brain. So estrogen is considered like a female hormone, but actually estrogen and testosterone are present in both men and women. There's just more estrogen and less testosterone in women, more testosterone, less estrogen in men. In the male brain, testosterone is converted to estrogen, and the estrogen is actually masculinized in the brain. In the female brain, there's an enzyme that blocks the effect of estrogen on the brain. This is why estrogen doesn't masculinize female brains. There are two disorders that have told us a lot about how hormones can affect the brain and later development. Androgen insensitivity syndrome is when an individual is a genetic male, so they have XY chromosomes, but they cannot respond to male hormones because of these chemical abnormalities. And from this, they actually develop a female phenotype. So because the male hormones cannot have this masculating effect on the brain and the body, they present as a woman. Androgenital syndrome, on the other hand, is when a female fetus, an XX fetus, is exposed to male hormones for a variety of reasons. And this can happen depending on the amount of male hormones that they're exposed to. More severe cases can result in masculinization of the genitalia as well. The effects of sex hormones on the adult brain are called activating effects because they activate the brain to do certain things. The hypothalamus controls a lot of the copulatory behaviors in both sexes, so like mating behaviors. Female sexual behavior tends to vary with menstrual cycles. Note, this is a deviation from the book. The book says that sexual behavior in humans is not does not vary based on female hormones, and this I have to respectfully disagree with this based on research. There's plenty of research that as ovarian hormones vary, there's increased sexual activity. So during ovulation, specifically in women, there's an increase in progesterone and estrogen and a bump in testosterone, and there's increased sexual behavior in women. It is a complex interaction, the female sexual behavior and hormones, and it varies a lot between individuals. There's also not been a ton of research on it. In males, it's pretty clear that testosterone regulates sexual behavior. It acts on the hypothalamus to produce copulatory behavior, and it acts on the amygdala to seek sexual activity. So there are definitely sexual differences there are differences in the sexes in how the brain affects sexual behavior. Sexual orientation and sexual identity are also affected by brain organization and hormones. So sexual orientation, just to make sure we're all using the same terminology, is a pattern of sexual attraction. This includes homosexual, bisexual, heterosexual, pansexual, asexual, queer. It seems like there's more every day, which is great. People can identify however they feel is most appropriate. And this is actually consistent. People tend to be more of a spectrum than fitting into one category necessarily. So at my alma mater, my undergraduate institution in Indiana, there's the Kinsey Research Center, and Kinsey came up with this scale. I think it's a 1 to 7 scale, where on one end is homosexuality, on one end is heterosexuality, and people actually tend to be somewhere in between, not necessarily a 1 or a 7. Non-heterosexual behavior is seen across different animal species, non-human animal species. There's this famous case, I can't remember their names anymore, of these 2 penguins that were at the Central Park Zoo in New York, and they were male penguins who were engaging in all of the mating behaviors and sort of what we would anthropomorphize as affectionate behaviors. And they paired up, and they even tried to, there was a rock that they thought was an egg, and tried to raise it. And the male penguins were eventually given an egg of a couple who was not successful in raising raising eggs. And they had a little baby together. The male penguins had a little, raised a baby together named Tango. And there's a lot of fan art on the internet about Tango and Tango's 2 male daddies, if you want to reach into that particular corner of the internet. But the existence of non-heterosexual behavior in animal species does indicate that there's, just even without any of the studies that we have, indicates that sexual orientation is in large part influenced by biology. And so there are both genetic and prenatal influences. In homosexual men, there are more feminized functional and anatomical brains in general. There's a feminized 2 to 4 digit ratio, which I believe I mentioned before in a previous lecture, where the ratio of the 2nd and 4th digits, so the pointer finger and the ring finger, is associated with prenatal hormone exposure. And so heterosexual women tend to have equal length in the 2nd and 4th digit. Heterosexual men tend to have a longer ring finger and a shorter pointer finger. And homosexual men tend to have the opposite pattern, so indicating less prenatal testosterone exposure. Homosexual women, like purely homosexual women, not bisexual women, tend to have a 2 to 4 digit ratio that's closer to heterosexual men. There are other marked abnormalities that this next one actually has not been explained in a satisfactory way by research, but it's interesting, and it's been observed in several studies that left-handedness is more common in homosexual females, quite a bit more common. I want to say at least 2-fold, possibly more. and it's slightly more common in homosexual males, although not as much as in homosexual females. And there's a lot of left-handedness indicates differences in brain development. It's not an abnormality, but it's just it's associated with differences in brain development as opposed to right-handedness, which is much more common. Ambidexterity may also be more common in bisexual individuals. And then there's this fraternal birth order effect, where men are more likely to be homosexual if they have more older brothers. There's the mechanism for this that's been proposed is that there's a possible immune response in the mother that maybe inhibits exposure to testosterone and decreases the estrogen's masculinizing effect on the brain. That's one possible reason for it. There hasn't been a ton of work on it, and of course in humans and prenatal environments it's really hard to do that type of research. But this fraternal birth order effect may account for, I think, something like 1 in 7 adult homosexual men. Finally, and I think this goes without saying, but there's no evidence that adverse environmental effects such as sexual trauma, parenting, affect sexual orientation. Men and or same-sex couples who raise children, their children do not have a higher prevalence of being homosexual as compared to heterosexual couples who raise children. Okay, and then there's gender identity, which is your personal affiliation with a given gender category. 2 categories include transgender, who is a person with a gender identity that differs from the gender they were assigned at birth. So someone who is born a man, but feels that they are a woman, that they're actually a woman. And then cisgender is a person with gender identity matching gender assigned at birth, so a woman who feels that she is a woman. So that would be probably the majority of the population. There are also other categories: genderqueer, non-binary, third gender. In Hawaiian culture, this is mahu, and a lot of different cultures have third genders. Exposure to prenatal hormones appear to play a role in gender identity. Some similar processes to different sexualities. Research is very preliminary. It can be difficult to get an adequate sample of individuals who are transgender or third gender, but functional and anatomical brain patterns often appear to match the gender with which the person identifies rather than the one they were assigned at birth. Okay, and so finally, reward circuit. The reward system, of course, drives behavior. We've talked about the reward system previously when we were talking about dopaminergic pathways and then drug addiction. So reward system drives a lot of basic behaviors: eating, sex, drug addiction. This is mostly the mesolimbic dopamine pathway, which mesolimbic is actually the projections from the ventral tegmental area, the VTA, to the nucleus accumbens in the basal ganglia. And this is part of the medial forebrain bundle, if you remember, which goes through the hypothalamus as well, and has a large role in driving motivated behavior. The mesocortical dopamine pathway is also associated with reward. This is dopamine projections, again starting at the VTA, but this time projecting to the prefrontal cortex, especially the dorsolateral prefrontal cortex. This is likely responsible for cognitive control, motivation, emotional responses. Both of these pathways involve dopamine transmission when an individual is engaged in the rewarding behavior. There are 2 components of reward. We touched on this previously in the when we were talking about drugs. There's the incentive, the wanting, the desire to use the drug, and then there's the liking, which is the pleasurable experience from using the drug. And these are rather dissociated, especially with drug use. Liking can decrease with drug use while the incentive remains. So people who are addicted to drugs often have that incredibly strong desire to use the drugs, the strong wanting, but the liking, the pleasurable experience has significantly decreased. Rats with lesions to the mesolimbic reward system seem to like food despite not actually seeking it out. So if it's given to them, they show these behavioral signs of enjoying the food, but they don't actually seek out the food. So the conclusion from this and other bits of evidence is that wanting is more associated with the mesolimbic pathway as opposed to liking. These were not mentioned in your book, or a lot of this information wasn't mentioned in your book, but I wanted to throw in some information about psychological disorders and psychiatric disorders that are associated with emotion and motivation. Major depression, which is an extreme and persistent sadness, and or emotional numbness. So there are two ways that people typically experience depression, and that's to be sad, to have like negative emotions. And then there's just sometimes people are numb. So it's the lack of positive emotions. People say they feel like they can't feel anything. And this is called anhedonia. We don't know exactly what underlies depression. It's likely that there are a range of subtypes with different neurobiological signatures. Certainly the feeling of extreme sadness versus not feeling anything, there's probably different neural circuits and brain processes underlying those two symptoms. But major theories do include the monoamine hypothesis, which is a little bit old and slightly outdated. At least it's at least more complex than what we used to think. But the monoamine hypothesis indicates that decreased amounts of serotonin, dopamine, norepinephrine, the monoamines, there are decreased amounts in some areas of the brain. And this would be why serotonin reuptake inhibitors are used for depression, or SNRIs, serotonin and norepinephrine reuptake inhibitors. These increase the monoamines in the brain to give an antidepressant effect. The limbic cortical model is a more recent model, and there are a lot of variations of it, but it says that too much activity in some limbic regions are associated with depression, whereas there's too little activity in prefrontal regions. So in those like emotional areas of the brain, too much activity, and then in the prefrontal areas where you're kind of able to control the sadness, control motivation, there's decrease. In bipolar disorder, again, really complex, not enough research, and we don't know enough about it, but mania is associated with bipolar disorder, which is prolonged euphoric happiness and excitability that's actually so excitable, sometimes it can be more irritation, or people do reckless things and can't concentrate, can't focus their energy. Bipolar disorder appears to maybe be like too little serotonin and too much epinephrine during mania. People with bipolar disorder often have the depressed state as well, but sometimes some people don't. But most people with bipolar also have depressed states, which would look more like the previous neurobiological profile of depression that I just mentioned. And then in drug addiction, as we've talked about before, there's a dysregulation of the reward pathways, especially the mesolimbic system. Schizophrenia, I would consider to be an emotional and motivational disorder. Depression and mania both exist often in schizophrenia and share a lot of the neurobiological signatures of those two disorders. So schizophrenia, there's emotional and motivational instability. There can be emotional numbness or lack of motivation, which is associated with hypoactivity in the frontal areas, just like in depression. Or there can be affect lability, which is an intensity and variability in emotions, so just kind of a lack of control over emotions, and this is again associated with limbic hyperactivity. There are other things that go on to that create the psychotic symptoms of schizophrenia, but this is some explanations that have been given for the emotional and motivational deficits in schizophrenia. And then anxiety. There's a range of anxiety disorders, general anxiety, PTSD, phobias, etc. But they're almost all associated with an excessive fear or worry. And the HPA axis, the hypothalamic pituitary adrenal axis that I mentioned before, is highly implicated in anxiety disorders, as well as an overactive amygdala in response to emotional stimuli.